DNA, the molecule of life. So we're looking at the learning intention, understanding how DNA structure was discovered, and it was discovered using new technologies, modeling, and collaboration between different scientists. So today we're going to concentrate in this video on the modeling of the structure of DNA and the roles of different scientists in actually discovering the structure of DNA. And the scientists we're going to focus on in particular are Rosalind Franklin, James Watson, and Francis Crick. So the two most famous discoverers of DNA are James Watson and Francis Crick, and they published their knowledge of the structure of DNA as being a double helix held together by bases in 1953. And they didn't come up with this idea all on their own. They actually built upon the knowledge of scientists that came before them. And the technique they used to see if they were right was to model the structure and see if it fitted all the available knowledge that the scientists had. And they actually used just cardboard and metal to actually model the 3D structure, which seems like a very simple process. And what they did was they put together all the sort of puzzle pieces, not only the literal puzzle pieces of different pieces of cardboard, but also the scientific puzzle pieces that other scientists had come up with. So that was the knowledge of how the bases, the A's, the T's, the C's, and the G's actually bonded together. They also used some new technological ideas. So they used work that was collected using X-ray crystallography. That work was done at King's College in London, and Watson and Crick were working in Cambridge, so they had to collaborate with their colleagues in London to get the data from the X-ray crystallography work. And they also used knowledge from scientists about how chemicals actually bond together. So all these different puzzle pieces came together when they were putting their model together to get it right. And they actually earned the Nobel Prize. This was so groundbreaking and so important that they won the Nobel Prize for Physiology and Medicine in 1962. And this was a Nobel Prize awarded actually to 3 scientists, and the third scientist that was credited was a man called Maurice Wilkins, because he was the one who provided the information from King's College London, which was the X-ray photos of DNA, which were pivotal in helping Watson and Crick to come to their conclusions of how DNA was structured. But there is a third person, and this is Rosalind Franklin. And Rosalind Franklin was a colleague of Maurice Wilkins, and she actually worked with her student to develop photos using this new technique of X-ray crystallography. And this photo in particular, which is called photo 53, is the one which allowed Watson and Crick to look at this picture, and they saw this X pattern in the center, and that was the information that told them that it was a double helix. And from this picture they could measure the spacing of how far apart each of the units of the helix were in terms of how twisted it was and what the repetitions of the base pairs was, so that they could actually build their model using some data. But unfortunately, Franklin isn't one of the people who won a Nobel Prize. And this was because she actually passed away 4 years before the Nobel Prize was awarded. And they don't give Nobel Prizes to people who are not alive. So she wasn't credited with any of her work for getting that photo. And she was also actually painted in quite a negative light when James Watson wrote his book on the history of the discovery of DNA. He didn't treat her with kind of professional respect. So this has led to quite a lot of controversy about whether they actually sort of cheated in their acquisition of that knowledge from Rosalind Franklin. and whether, because they didn't credit her, was that really a successful way to collaborate as scientists. I will leave you to think about that ethical conundrum yourselves as you explore how new technologies, collaboration, and modelling were used to discover the structure of DNA.